Save. Okay, it's recording. All right. So with this class, I'll teach it once a year. So I have like another computer with me so I can see my notes. So if you see me kind of look over and have to reference something, that's why. Um, just I don't teach it all the time. Okay. So I'm gonna record these lectures, ask questions as needed. And if this time doesn't work out for the group, we can figure a different time that works better for people. Um, but yeah, I'll record it, I'll post it so you guys can have that. So, little refresher on prokaryotic versus eukaryotic. So, prokaryotic, unicellular, no nucleus, um, smaller in size, no histones, only ribosomes as far as organelles, versus eukaryotic, typically multicellular, have a nucleus, membrane bound organelles, so all the Golgi, mitochondria, all of that good stuff. We in our cells 46 chromosomes, in theirs one circular chromosome. Okay. So as far as organelles and function of cells, so something we kind of touched on. I know I touch on it in my bio 1111 class. I can't remember if AMP1 touches on it. Do you guys remember what the purpose of smooth ER is?
It's synthesis, right?
Synthesis of lipids.
Lipids, yeah.
Anything else? Carbs.
Probably.
Storage of calcium. That's your endoplasmic reticulum or sarcoplasmic reticulum in your muscle. And then lastly, detoxification. So this is often referred to as the cytochrome P450 system. And interestingly enough, like when I went through school, that's all we talked about. CYP 450, CYP. Now there's all these subsystems. And what's interesting with it is this system can upregulate. So there's a lot in your liver. It'll upregulate if you eat grapefruit, drink grapefruit juice. And that's why, like, on certain medications, you'll see, especially like, for example, oral contraception, you'll see do not consume grapefruit juice or grapefruit with it because it upregulates the system. It breaks down the drug more rapidly, and the drug is non-effective. If if the pharmacist does not educate you on this point, you know when you pick up your meds and they're like sign here, that's technically when they're supposed to educate you. Um, if they do not, and you take oral contraception and you drink grapefruit juice, you get pregnant, the pharmacist will be liable for the child for 18 years. They will pay child support for the child.
No way.
Yeah. So that's why they just have everyone just sign, sign, sign. But you are entitled to that counseling. Like, doesn't matter what they're in the middle of, they should counsel you if you want it on a medication. Okay. So what's interesting too with these subsystems, like my wife deals with this, they will test for it because it affects the different breakdown, like different drugs are broken down by these different subsystems. So if you're like very high in one of these subsystems, you'll break down the drug very rapidly. They need to give you more drug or like dose it sooner than the average patient, or vice versa. If you have a deficiency in the system, the drug will reach, you know, appropriate levels much more rapidly. Um, so yeah, it's pretty neat that they're discovering this and even like dosing off of it, like pre-testing the patient. It's expensive to do, so it hasn't really caught on yet. It's in that shifting stage, but they'll pre-test the patient and it affects like painkillers, for example. So they can dose based on that and how much will be in their system and give them a more appropriate dose. So pretty neat. Pretty neat that they can do that. So there is a disease that affects the lysosome system that I have in my notes here that they talk about, and it's called eye cell disease. So they're deficient in the function of the lysosomes, and it'll release the lysozyme into the blood, and it actually can damage other cells. It can actually create damaging throughout the cell system, throughout the body. Pretty rare disease, though. Okay. Cell membrane. So we have cholesterol in our cell membrane. Uh, signified here. And that helps with fluidity. So when it's like super cold, it doesn't fracture. When it's super hot, it doesn't lose its integrity and fall apart. So cholesterol is a necessity for our diet. Fat is a necessity for our diet. Yes, we can make it, but we do need fat. If you ever seen like when they show like those ads that are like, donate, these kids are starving in Africa, and they show the kids and they got these big old bellies. The reason they have those big old bellies is lack of fat in their diet. They're getting rice. That's pretty much what happens. Rice is cheap, it's filling. So they're eating just straight carbs. And so they develop what's called kwashiorkor. And what happens is when their liver is packaging I think it's VLDL, very low density lipoprotein, it can't exit from the cell, and so the liver starts to grow. What you're seeing with that protruded belly is their liver. What's wild is we're seeing it here in the US. There are cases of kwashiorkor in college students. They're saving their money so they can buy alcohol and instead are eating ramen. It's like the cheapest thing to eat and it's filling, right? So it's just wild that we're seeing kwashiorkor here in what wealthiest country in the world, probably. Okay. All right. Little refresher on cell membrane proteins. So you guys learn about cell recognition. It's how cells recognize cells. Cell communication, transmembrane proteins, receptors. Um, there's growth regulators. What else? There's like 8. Storage of storage for energy. But yeah. So the reason these proteins are important when you talk about like antigens, for example, if your immune system misidentifies that surface antigen, that surface protein, what do you end up with?
Autoimmune disease?
Autoimmune disease. It will attack it, it will think it's foreign. The receptors are necessary for medications sometimes. Certain drugs will bind to it. And then even in ourselves, if you're deficient in certain receptors, certain systems won't downplay. So, for example, like people that have genetically high cholesterol, hey, um, I probably have deficient receptors or different or production of the hormone that communicates with that receptor, and so my body overproduces LDL. Like I rarely eat fried food. Yeah, I eat red meat. Okay, I love my steak. But my cholesterol is astronomically high. I think last time or before I got on my most recent like round of medications, my LDL was like in the 230s.
Oh,
It
wow.
should be below 100. That is astronomically high. So now I'm on an injectable. It is a antibody, immunoglobulin, uh, IgG, right? And so it's an injectable I take every 2 weeks. My LDL on the last check, and it's probably dropped since then, was 69.
Oh, wow. That's
Yeah,
great.
it's a heck of strong. Inject every 2 weeks, but it works, right?
Hmm.
So these are a lot of these new medications that are coming out, that are changing the game, really. So, transmembrane proteins, this little simple structure, if it's messed up, this is cystic fibrosis. If it's messed up, and we're messing up one nucleotide basically in the DNA, which changes one nucleotide in the RNA, which changes one amino acid in the protein structure. That's all we're changing. One amino acid in the end. What happens in cystic fibrosis, they cannot secrete chloride across the membrane. So say we're in the lungs here. This is like inside the alveolus. Because chloride does not go in there, water doesn't follow. And so they create this thick viscous mucus. So this is why they wear like those vibrating like vests now. They used to like beat on their back to like break up the mucus, so they can cough it up so they can breathe. It inhibits their ability to diffuse oxygen and carbon dioxide. It also happens in the pancreas. So you'll see like they don't secrete like all the lipase, amylase, all that good stuff, so they can't break down food. So even though they're eating, they're not absorbing the food, and so they can have failure to thrive, for example. Pretty devastating disease. Um, Extracellular matrix. So here we're talking about like integrins. These are attachment points so that cells aren't just floating around per se. Cells can't replicate if they're just floating around. This will help, like, so regulation of growth, for example. And they mention here, like in the notes, fibronectin is one of the big guys that plays the role in here for adhesion, growth, migration, differentiation of cells. Cell junctions, so attachment of cells, creating tight barriers, so like moisture can't get through, bacteria can't get through. Also for communication between cells, transfer of electricity in the heart, transfer of nutrition cell to cell. So all of those are important functions there. Signaling. So with signaling, here's endocrine. So we're releasing a hormone into the bloodstream, targeting somewhere else in the body. Must have the receptor for it. Paracrine, so this is on neighboring cells. So this would be like beta and alpha cells in the pancreas. When beta cells release insulin, the insulin turns off the alpha cells, which release glucagon, and vice versa. Autocrine, this is on the cell itself. So it will target itself, possibly block like an entry point, something like that, shut down protein receptors, depending on what's being secreted. So it would be like a uh, negative feedback system or positive feedback system, even. All right, cellular respiration. Do you guys remember it? Just even in layman's terms. I don't need all the numbers.
Oh my goodness.
So sugar is broken down into pyruvate, goes into Krebs cycle, and then turns into ATP in the mitochondria.
Okay. And what's the actual like equation?
Oh man. What's the
Glucose plus.
Is it glucose plus
Glucose
oh
plus
man.
water, right?
Is it
No,
is it
CO2.
Do you breathe in CO2?
No,
so is it C6?
oxygen.
Oxygen. Glucose
There we
plus
go.
oxygen. Arrow. What comes out the other end? What do we breathe out?
The carbon dioxide.
Carbon dioxide. What else?
Um
Cold day,
Water. Is
water,
it
and
plus
then
ATP,
and
right?
ATP. And how much ATP do we make?
Like in a okay, it's around like is it 30 something?
30 something is
38?
38. Depends on your textbook. I've seen anywhere from 32 to 38. I teach it in four classes, technically, including this
Hmm.
one, and every book is a little different.
Okay.
It's somewhere between 32 to 38 ATP per glucose molecule. It's a um, very inefficient system because 66% is, I think 66% is released as heat, 34% is ATP. It could be vice versa. Regardless, it's inefficient. We do need the heat because that's how we thermoregulate. Um, but yeah, I have in here, we have enough glycogen storage for 1 day, enough fat for a month or more. I'm on the more end. So, what happens if you don't have oxygen?
You die.
Then if what
Yes, but
without
if
yeah,
locally at a muscle, let's say there's not high oxygen load at the muscle, what is it going to do instead of cellular respiration?
it's gonna go to glycolysis.
It's gonna
Yes.
fermentate.
It will go through
Fermentation.
glycolysis and then to fermentation. And how much ATP do we get from that?
Two.
Two.
Two. Very inefficient system. When we talk about like cancer, cancer cells prefer that. And
Mm-hmm.
so they rapidly eat through nutrition. And so that's why you see like when someone's like terminal with cancer, they go through what's
Mm-hmm.
called cachexia. They
The
lose weight
catch,
like
yeah.
wildfire. Doesn't matter how much you feed them, They're the consumption rate by the metabolic rate of these cancer cells is just
Hmm.
through the roof. They're burning through thousands of calories a day.
So is that how like I've always like had like how do like cancer patients like really actually die? I know it's like, the chemotherapy, but how do they like what goes on in the body? Is it because of what is it called?
Cachexia.
Cachexia?
Cachexia.
Is
Yeah.
it because of cachexia or is it because there's so many
It
like
could
things in the
be.
body?
It depends on where the
Yeah.
cancer is, right? Like,
Mm-hmm.
could they starve to death? Yes. And so
Mm-hmm.
the body fatigues over time, like the muscles aren't getting nutrition, the heart isn't getting nutrition, and fatigue stops eventually. Yes, that
Mm-hmm.
is a possibility. But if you have
Okay.
like brain cancer and the tumor grows and grows and compresses the brain, the functionality
Mm-hmm.
of the brain ceases to exist. You have
Right.
obstruction of vessels as it multiplies inside vessels. It just destroys the tissue. So it really
Gotcha.
depends. Like you have liver
Okay.
cancer, it destroys the liver. You can't function without the liver because you don't have the
Right.
detoxified.
Exactly.
Yeah, it depends on where the cancer is.
Gotcha. Okay.
But yeah, one of the signs of cancer always is sudden weight loss that is not intentional. And
Okay.
that's late cancer usually.
Perfect. Okay. Cool. Thank you.
Welcome. So cell transport, this is, where we talk about isotonic, no net flow. It's equal side to side.
Mm-hmm.
If we have a hypertonic solution, the water will move over towards that solute. So if we had a lot of salt over here, water will follow. And if we have a hypotonic solution, water will leave and go towards the other side. All right, transporters, uniport, one, uno. Okay. Symporter just means two things are traveling together. So this is a co-transporter, and he
Mm-hmm.
is a symporter, meaning two things are traveling in the same direction. This is also a co-transporter, but it's an antiporter. They're moving in opposites. All right. So here's some questions for you. What is required to move water across the cell membrane?
Is it diffusion?
So when we talk about water, osmosis, diffusion is always
Oh,
solute.
It's
okay.
aquaporin, right? The
Aquaporin. So we have a little pathway where water just can pass in and out. No
Mm-hmm.
problem. Um, is water polar or non-polar?
It's
polar.
polar.
Polar. And that's why
Okay.
you need that transmembrane protein. It's just the channel to let it pass through. A very
Okay.
large protein, would you need a transport membrane, transport protein, membrane protein? Would you need a protein to let it go through or no?
No, I don't think you do.
A really large protein won't just diffuse across the membrane. It has to be brought in, so it'll like the membrane might over encompass it and bring it in, or other different things, but it takes energy.
It takes, yeah, well, it may not require energy, but it does require a transmembrane protein. If it's very
Mm-hmm.
large, it needs something. So remember, with the cell membrane, it's hydrophobic and
Mm-hmm.
it's tightly bound. So only small hydrophilic things can pass through. Okay.
Right.
Oxygen. Freely pass or no?
Yes.
Yes.
Yes. Think about like in the lungs. Do I need a transmembrane protein for oxygen to come into my body?
Nope.
Okay. Bacteria.
It seems to get
Mm.
in by itself just fine.
It actually triggers a receptor to enter or commands the cell to enter. It can't
Um.
just freely enter. So it's interesting because when you talk about like viruses too, same catch there. So if there are people that do not possess receptors for viruses, I don't know
Mhm.
that for bacteria that exists, but for viruses, they do not possess the entry receptor. The virus can't enter into their cells. They cannot be infected. It's pretty wild.
So
Just,
what? So for viruses, like, what is the like? Say for example, HIV,
mhm,
right? You can't. You get that, from anal sex, vaginal sex, needles, and everything like that.
mhm.
What is the how does it? I know HIV binds to a receptor.
Hmm.
It binds to a receptor. Is it that CD4 receptor that we have?
No, it's actually like
Oh,
I think it's CRX5, CXR5.
oh, okay.
Yeah,
It's a lot.
it's on the CD4 cell.
Oh, okay,
Uh,
okay.
yeah, it's on the CD4 cell. And that being said, that's one of the ones where they discovered that whole thing where some people are immune to viruses.
Mm-hmm.
It was a pretty neat case. The guy had like leukemia and HIV, and they did a bone marrow transplant on him, and all of a sudden his HIV started disappearing from his body,
Oh, wow.
and that's where they discovered it. I think it's CXXR5. And if you are homozygous, you're immune. If you're heterozygous, you're not immune. Uh,
Oh,
yeah, so the thought
okay.
process is with all viruses, that potential is there.
Okay, gotcha.
And then carbon dioxide, freely pass or no?
Yes.
Yes.
Yes. Think about it in the lungs, freely passes. Okay. Um, so this is going back to like the bacteria, for example, receptor mediated endocytosis. That's where the bacteria strikes a receptor and commands the cell to pull it in. Same with viruses, commands the cell to pull it in. LDL, same thing. So like in me, for example, I'm pretty sure I have familial hyperlipidemia. Something's going on with the receptor, and so my LDL from like age 18 on has just been rising, 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 no matter what diet, doesn't matter. Um, and so the thought process there is the receptor is non-responsive, something like that. And you can even say the same with like type 2 diabetes. That's where they talk about like insulin resistance. The
Mm-hmm.
receptor just may not be accommodating the insulin that is striking it. The person is making normal insulin, they're just not communicating. The fit isn't right for some reason. All right. So DNA, when it's in its spread out form, that's chromatin. As we condense it down, we use histones, these protein structures, to condense it. And then here we're looking at a duplicated chromosome ready to divide. Okay. So we need it in chromatin form to read it, to read those nucleotides. Little refresher on mitosis. So this would be interphase, prophase, metaphase, anaphase, telophase, two identical daughter cells to the original cell. We go through mitosis like crazy. There are checkpoints in this to regulate, the cell from going on. So that would be your um, P53 checkpoint, and then there's two other checkpoints that we talk about, like RB checkpoint, and it's checking that everything is correct, and I believe the M checkpoint during metaphase plate time to figure out that everything's lined up. And so we do this so that we don't develop cancer, because if a mutated cell goes through this process, that's it. Now it's going to replicate like wildfire with that mutation. Some cells remain in what's called G0. They do not replicate. So, like heart does not replicate, brain, neurons does not replicate, skeletal muscle does not replicate. So, once you damage it, that's it. It can't repair, it can't replace. And then some cells remain in the mitotic, like G1, encircling in that whole signaling process and replication process, like your hair, your nails. They're constantly skin, constantly replacing, replenishing. So this is talking about that RB gene, the retinoblastoma. Retinoblastoma protein. That's that inactivation gene. So like growth factor has to remove it or inactivate it so that replication can continue on. If there's a mutation in this, it's typically in the heart and eye, and it creates a true heart tumor. It's like a growth, and it's kind of neat when it creates like a little pendulum, like a ball hanging. And so the patient, it'll clog I want to say it's the aorta exit from the left ventricle out to the aorta, and it'll block it. And it's like if they lean certain ways, it won't block it, and they'll feel fine. If they lean other ways, it'll block it, and they're not getting proper circulation. And then it causes a tumor in the eye. And so in the eye, it'll cause like a one-eyed, like red eye, essentially. And so Parents usually spot it in the child when they take a picture of them, and it bounces back this weirdness in the eye. That's usually how they spot it, and it's a defect in the RB or retinoblastoma protein. Growth factors. So this is talking about um, when they're overexpressed, we can see cancers. So like certain breast cancers are what are considered HER2 positive, and we've actually created drugs against these. You may have seen them on TV. They advertise them from time to time. Um, all these different growth factors, and they can regulate it that way and turn it off and turn the tumors off essentially, which is pretty neat, or at least slow the growth to where they can deal with it. Transcription translation. So this is where we're making messenger RNA. That's transcription inside the nucleus. It then goes out to the ribosomes to be translated into the protein. Genes, those are just inheritable characteristics on chromosomes. Um, when we talk about genetic diseases, either we don't pass on the gene, and so we have a deficiency there, we're missing it, or we pass on extra genes, so like trisomies, like Down syndrome, or it gets mutated and it's faulty. So that would be like cystic fibrosis that we were talking about. Lacking genes, you can miss a whole chromosome even. So that's your monosomy, so like Turner syndrome, for example. A little refresher on proteins here. So we have the primary structure. That's just where it's in a string of amino acids. Then we can the secondary structure. We start to fold it. Uh, it can be an alpha helix or a beta pleated sheet. That's kind of like where it's folded back and forth. You know how you used to make like a fan in elementary school where you fold the paper piece back and forth? And then it goes into the tertiary where it actually folds. And then quaternary is where we take four of those tertiaries and put them together. That is the complex structure. All right. So that's the first lecture. And we'll move on to the second one. If I can get to the thing here. There we go. And the second one is what? Altered, I believe. Cell alteration and injury. Let me just confirm. No, genetics. Altered is next week. Where is genetics? Ah, there it is. Hmm. All right. Is it showing up for you guys full screen?
Yeah.
Cool. Cool. All right. So, genetic diseases. So, the DNA code can be altered in any way. It could just be random. It could be because of mutagens, carcinogens, age, any of that. So, what happens is either we delete, a gene, we're missing information. We add a gene, now there's conflicting information, or we add or delete entire chromosomes. So you can see any of those happening in that situation. So what triggers it? Like I was saying, mutagens. Um, those mutations are passed to the offspring. And so what's the end result? Basically this entire class, potentially. Whatever disease can be seen down the road. Okay? We can't repair the errors. I have here in my notes that spontaneous mutations happen 1 in 10,000 mutations. Or, sorry, 1 in 10,000 people. A random spontaneous mutation can happen. No, 1 in 10 million. Sorry. Okay. There's fewer than 1 in 1,000 base changes in the DNA, which result in that permanent mutation. So it's a pretty safe system, but it can happen. We are able to repair DNA. The problem is it's just going to slap whatever it wants in there. If you guys remember, I think it's on, yeah, on our next screen, the redundancy in here. So, like, for example, let's say we accidentally swap out that U right here, we still end up with serine. We put a CAG, we still get serine. So the redundancy helps prevent that. Um, it's called wobble, codon wobble. That's what the term is for when that third guy accidentally gets swapped out. Now, that being said, just because it swaps out, the protein could still be semi-functional. It could kind of work, and that's how you get variants in disease sometimes, like a spectrum of disease, depending on how severe, depending on where in the chain it happened as well. That can have an effect there. I have in my notes here that 1 in 12 conceptions have a chromosome issue, and that's why one of the reasons women are encouraged to not share really or that they're pregnant during the first trimester because it can self-abort at any time because of a chromosomal error. So it does happen. Okay. First, we look at base pair substitution here. So we have CUC, which should be, where's CUC? There it is, leucine. And we change it to CUA. It's still leucine. This is a point mutation, and it's a silent mutation. No harm, no foul. These happen. You wouldn't know unless someone took your DNA, had it from the very beginning, and then later in life checked it, and they go, oh, look, it changed. Nothing to worry about here. Here, we have an amino acid change. So this is going to be a missense. So here, this is what it should be. Here's what's changed. Here's what we end up with. So we went from glutamate to valine. That's a missense. Now it is a non-properly functioning protein. Sickle cell is a rough disease. It's incredibly painful. They have sickle crisis. Uh, it occludes vessels. They lose their spleen by like age 13 or so. And so, because of that, they're susceptible to encapsulated bacteria infections. The memory tool for that is Shin, that you have to vaccinate them against. So that's strep pneumo, Haemophilus influenza, Neisseria meningitidis. So they should be vaccinated against that. Cool thing is, they're immune to malaria. Malaria needs to infect the red blood cell and then take the membrane because it's all dysfunctional. The malaria can't survive in them. Also, people with sickle trait, they're carriers. They do not sickle. They're also immune to malaria. It's pretty neat.
Well, that seems to be where it's at. Like, get the benefit, not the drawback.
Yeah, and it's interesting when you think about it too, like this is kind of like an evolution, right? Where malaria is most predominant is in Africa, and that's where we see sickle cell coming out of. Probably what happened at some point, someone had a mutation, they had the immunity, their genes were now being populated the most because they're surviving the malaria infections. So it's pretty cool. Pretty cool.
So they're surviving the malaria infections, but they're not necessarily surviving to adulthood. Like, so how did they spread this gene on so well that it's
The,
commonplace?
the carriers. The
Oh.
carriers are who's really passing it on and surviving no problem. I mean, they are like superhuman, right? Like they don't have the sickle crisis, and then get bit by a mosquito, no problem, no malaria, move along. So their genes survived. Survival of the fittest, right? Um, Here, when we changed over and we put in UGA here, UGA is a stop codon. Now this does not code. The protein is truncated. This is called a nonsense mutation. Depending on where it happens in the protein, will differ how the disease is seen clinically. If it's real late in the protein code, it's not going to be really severe. Mild symptoms. If it's real early on, severe symptoms, right? The complete protein is non-functional. Frame shift. This is a deletion. Because we delete like this U here, remember codons are threes. Everything downstream is changed. Once again, where this happens, if it's very end of protein, not severe. Very early on, severe. The entire protein is something else. So that's frame shift deletion. There is also frame shift insertion. It would have an extra. Same thing. Everything downstream altered. So here, cystic fibrosis is a deletion. And because of that, it changes the functionality of the protein. It can't pump that chloride ion across the membrane. Water can't follow, thick mucus. Here in my notes, I have 1 in 3,000 babies have it, highest in the Caucasian population. So commonly you'll see that when we talk throughout pathology, that certain populations you'll see more diseases of. That's very common. It's in their genes, and as they mix in their own population, you see more occurrence of the disease because we pass on one chromosome from mother, one chromosome from father. We have two alleles in us. We use one or the other. So when you come from the same population, like your parents are the same background, there is increased risk that they're both carriers of a disease or that they're passing on a defective chromosome or a defective gene to you. And so the probability increases as you mix in your own population or own background, if you will. There's increased risk of disease happening, and we even see it in like an entire population. Ashkenazi Jews, they hail from like the Black Forest area, like Eastern Europe, and they have tons of genetic disorders because they do not um, mate with people outside of their group. And so,
Oh, kind of like Tay-Sachs disease, right?
yeah, Tay-Sachs is one of them. You see a lot of Tay-Sachs in there. Um, oh man, I forget the other ones. There's quite a few. So like Tay-Sachs is the one where they have the cherry macula,
Hmm.
and I think they can't break down something.
They're like the blue people of Kentucky that they were isolated and they interbred for so long that they literally turned blue and they couldn't figure out why because
Yeah,
they
that
kept thinking it was oxygen, but it
it was
wasn't.
like the methemoglobinemia or whatever. It would build up in their tissue. They couldn't process something. So yeah, you see a lot of defects in there. Literally in med school, we learn about that population because of that. And it's funny because I was talking about it in my bio class Wednesday, that same exact point. And one of my students, he's Ashkenazi Jew, and I was telling my one of my sister's friends, he is also, and he had spontaneous pneumothorax like 3 or 4 times by the age of like 20 or 21.
Oh, wow.
This student said same thing. He had had quite a few spontaneous pneumothorax, so it's something in their genes where the lungs have like blebs in them, like these outpouchings that are weak, and it'll just spontaneously rupture. So it's just a genetic disorder. So frame shift insertion, like I was saying, something gets added in, and so all downstream it is altered. Once again, depending on where it happens, alters how much or how affected the protein structure is. Deletions, duplications, and translocations. So translocations is where two chromosomes, and one we typically talk about is 9 and 22. Chromosome 9 and 22 shift portions of their chromosomes onto each other. Uh, it's not appropriate because now the genes are in the wrong location. And so that one is referred to as the Philadelphia chromosome, and it causes a one type of leukemia to be very predominant in that population. Inversions, so they can be flipped on themselves, and then repeats, trinucleotide repeats. So, for example, they're talking about here CAG repeats. So, if you have a lot of these repeats, you are at high risk of Huntington's disease. Huntington's disease is a form of dementia that exhibits what's called chorea. Their arms, legs will randomly flail around. They cannot control them. If you guys watched House, And you remember the
I
girl,
love that show.
remember number 13 at the end?
Yes.
And she was worried about developing dementia because her mom had it. That was
Hmm.
Huntington's. So
Oh.
the disease is kind of unique to these trinucleotide repeat diseases. They exhibit what's called anticipation. So if like the parent has it and they start exhibiting Huntington's at 45, the offspring will have it at a younger age, like 40. Their offspring would have it at a younger age and then younger and younger. So theoretically, these would disappear over time because they're going to happen so early and it's before, they could introduce their genetic or genome into the population theoretically. Okay. Mm, yeah, that's all we need to talk about in there. Fragile X. So here with Fragile X, the males tend to exhibit it. They have a repeat of CGG. So it's on the long X of the chromosome. It can actually break off. I have in my notes 1 in 4,000 males has it, 1 in 8,000 females. So they have deficits in IQ. They tend to have like their ears are a little pointy. My old roommate, his brother had it, and he was decently functional. He would like go to school, he'd jump on the computer and search stuff. He could talk to you, but he had probably the IQ of about a 5 or 6 year old. Okay. That's about where he was. And at the time, he was like 20, 21, right? That's as much as his IQ is probably going to develop. Right. Oh, and they also have like their arch of their foot is usually very high. That's another sign that they have Fragile X. So you got to remember on like every chromosome, it codes for different areas in the body, and that's why you see these different effects, even though you're just messing up one chromosome all throughout the body. Here's translocation, so that's that Philadelphia gene, so the 9 and 22 swap, and it's the specific leukemia is CML, which is chronic myelogenous leukemia. There's four leukemias. There's AML, ALL, CML, CLL. So CML, I believe, strikes around the 40 to 60 age range. It's typically where we see it. Pretty aggressive leukemia that needs to be dealt with, or tough to deal with, I should say. Okay? But I think they said, yeah, 95% of the people that have CML, when they test them, have this translocation. So it's very predominant causation of this specific leukemia. Okay. So why is it relevant? It's all these diseases that are caused by it. So we talked about sickle cell, color blindness, right? They can't see certain colors. Hemophilia is clotting disorder. They bleed out very easily, destroy joints, can have brain hemorrhaging, all kinds of things. Cystic fibrosis we talked about. Phenylketonuria. This is a neat one. It's called PKU, is the shorthand for it. That's why if you look on like if you drink like Coke Zero or something, when you look at the ingredients, it'll always say like contains phenylketonureics. Those are those synthetic sugars. It accumulates in them. They can't process it, and they can actually die from it. Everyone here in the U.S. is tested on day of birth for PKU, since we do see it. So they just need to avoid those foods that have that, and they'll be fine. Okay. Translocations. So we talked about CML, Burkitt's lymphoma. Um, this is really associated with EMB, Epstein Barr, EMB, Epstein, EBV, Epstein Barr virus, which causes, what does EBV cause? Y'all took micro, right?
Yeah, but I think it's like I thought it was a skin disease, but now I'm thinking maybe it wasn't.
Not a skin
Like
disease.
You said EBV?
EBV, Epstein Barr virus.
it's like a STD
Well,
maybe?
Mm-mm.
Hmm.
You
Epstein
probably have
Barr.
had it.
Epstein Barr.
Chicken
I've
pox?
heard it.
Okay.
That's
Is it herpes?
varicella zoster.
Herpes?
Mono? Mono?
No. Mono?
Mono.
Oh.
EBV, mono. So in this population, I know I show you guys Burkitt's lymphoma. That's where they have the big old jaw tumor typically. Um, they have to have this translocation and catch EBV, and then they're at risk of it. We really only see it over in Africa typically. You don't see it here in the US too often. Deletions, Duchenne's is a muscular dystrophy. So the muscles, the protein is defective. The child is normal initially, and then after a while, you'll see they go to get up off the floor. They'll kind of lock their legs straight, and then they push themselves up, and that's a sign of Duchenne's. The muscles deteriorate ascending, so it starts in the legs, climbs, and eventually they have to be they're wheelchair bound eventually, and then they have to be on like a vent to survive. Okay, Huntington's fragile. We talked about. All right. So, like I mentioned, you get two alleles. Here they're showing this would be during meiosis when prophase when we're going to cross over information. So these genes would cross over at this point. This is your tetrad, okay? And it's fine as long as we cross over these portions. The problem is when you cross over like with these two guys. These are two different allele or two different chromosomes, non-homologous chromosomes. Autosomes versus sex chromosomes. So autosomes are the first 22, sex chromosomes, the last guys here. So we can have anomalies in any of them. So for example, they have an anomaly here where they have an extra X chromosome. So it is an extra Y. I think this is an extra Y. I think this is XXY is what that is. Which I think doesn't exhibit any. No, this is meta female, maybe. This might be meta female XXY. I can't remember off the top of my head. But we have our handy book. Let's see, or we can just Google. That's gonna bother me. XXY. Let's see. XXY syndrome. Klinefelters. Sorry, it's Klinefelters. Klinefelters. So Klinefelters is typically they're very tall, slender. They always say they kind of look like a prepubescent boy. That's what they kind of reference it as. Um, they're sterile. They can't replicate. The ovaries are referred to as like streak gonads. They're not really there. Okay, so that's Klinefelters. Uh, but yeah, so moving along, there's all kinds of different anomalies throughout there. Okay, non-disjunction of sex chromosomes. So this is Klinefelters. That's what they're talking about. Sorry, it's testicular atrophy here, not streak gonads. But yeah, usually very tall, slender. Okay, that they talk about. They say 1 in 5,000. No, 1 in 500 males can have Klinefelters, right? Now, you can have female versions of this. It depends on how much the Y chromosome kicks in. So, initially, everyone is female, even if you're XY initially. Eventually, the SRY gene activates on the Y chromosome and it starts to turn off the X activation so the male characteristics can take over. So if it does not activate properly, like if they don't get a full Y or something like that, then they'll be more female in appearance. So it just depends on how much. What is the biggest factor for non-disjunction in general, not only of sex chromosomes, but autosomes? What's the biggest factor? What do you think is going to affect during meiosis splitting of the chromosomes? Any
Mm,
I
guesses?
I'm
mean,
not
it's
too
just
sure.
rang genetic mutations that, like, epigenetic stuff, like, I don't know, being exposed to radiation or
Could. But there's one that's like the biggest factor.
I have no clue.
Yeah, I don't know.
Maternal age. Maternal age.
Oh,
Oh.
At 35, it is a high risk pregnancy because that egg is 35, actually older than 35 years old, because the female forms them in utero. So as they age, the ability of them to divide appropriately starts to deteriorate. So maternal age is the biggest factor. So it's not surprising, we see more of these genetic disorders as we go on. Males play a role in it, but they say males not till they're like 60 or 65 years old because they're constantly making new sperm. So
um, so I
it's
read that
mhm.
more elderly women, when they conceive, their children have, messed up genetics or whatever, but since they're more likely to get genetic testing because they're older, it's actually in younger women that more
Like
goober babies
random.
are, yeah.
Yeah, like randomly, yes, I agree with you. And then in the older population, though, the risk of it happening is much higher. Like if they weren't doing genetic testing, right? If they're doing genetic testing, we know there's high risk. Or if they're doing in vitro fertilization, then they could test the egg, look at it, verify it's a good egg or not. Okay.
Well, I mean, over 35, they used to try to do amniotic sampling to test for stuff.
Well, now they're even doing they're getting away from amniotic sampling because it's dangerous. They're doing the blood test. There's like a new blood test where they can the fetus's chromosomes as the cells break up, the chromosomes migrate into the mother's bloodstream and they can test for them. It's not as accurate, but it's less invasive, less risk of rupturing like placenta, less risk of infection, all of that stuff. So they are shifting over to that. Unless I think the only time they really do amniotic testing anymore was like for Downs, to be sure of Downs. There's like a protein in there they test for. That may have changed though. But yeah, it's interesting, like as being a teacher and stuff and watching the classes over time, the shift to where it's more females in my class. I think even my AP2 right now, I have 19 in there, and I'm pretty sure it's all women. My lab is all women. And even talking like Dr. Casey, he's noticed it too. And so women are going to school more, pursuing careers, and so they're having children later, right? So it's not surprising that we see more and more genetic disorders, and it's interesting because you watch like that you read studies and stuff and they're like, well, this is the reason, this is the reason. You hear people saying like, well, it's all the fast food in our diet and all this, and it's like, is it really? It's that whole like, correlating information, like how do you prove this is the only variable that is causing whatever the issue is, right? um that's science for you. Okay. So, mitosis, when we replicate, we create two genetically identical daughter cells. Meiosis, we create four unique gametes. That is in the male. In the female, they just create one egg. The reason is they take the cytoplasm as they divide, they take the cytoplasm so that it can be large enough of a cell. So non-disjunction and autosomes, only a few are compatible with life. So here, this is Downs. Um, so the there's three that are compatible with life, and they are on chromosome 21, 18, 13. So on 21 is Downs, on 13 is Patau's, 18 is Edwards. With 13 and 18 though, they tend to have like they call them rocker bottom feet. Their feet, the arch is actually like that. And then I think Patels, they have like clenched hands. The hands aren't really functional. Okay. And then aneuploid is when they find a cell that just does not have all three chromosomes somehow. Okay. Somehow it has the regular chromosomes. Okay. Or wait, let me think about it. Yeah. No, it's where it has extra. Sorry, aneuploidy is where it has extra. It is not the appropriate number of chromosomes when you're talking about gametes, not the normal 23. So if it has like 24 or something like that. In my like stats here, it says women over 45, the chance of having a trisomy is 1 in 20. That is astronomical numbers. Turner's, this is a monosomy. They're missing either the second X or the Y. Okay, they're missing that sex chromosome, so they tend to be very short neck. They have this lymphedema here, so they call it like webbing. The nipples are very widely spaced. They're short, and then this is the ovaries are streak gonads. So back in the day, they didn't know, and the uterus is like a blind pouch, is what it's referred to, or the vagina is a blind pouch. There's no uterus. So they would, back in the day before we could do genetic testing and all that, they would like get married, try and have a kid, stuff like that, and they couldn't get pregnant. And then when the vagina was inspected, they would see it's just a pocket. It doesn't go anywhere. Um, I always think of, did you guys ever watch Mad TV back in the day?
Yeah, way back in the day.
Do you remember It's Pat? Where they
No.
can, it was It's Pat was a character on there, and it would always put people in like these awkward situations, like you didn't know if it was a guy or a girl. And I always think of that when I think of Turner's. Go look up It's Pat. You'll see what I'm talking about. All right. So recessive and dominant diseases. So recessive diseases, the parents both must be carriers. Okay. If they're both carriers and they pass on that gene, each of them, the offspring can have the disease. If they pass on just one good gene and one bad gene into the offspring is a carrier. Or they can pass off both good genes and there's no disease, no carrier, it's gone from that genome. Okay? Dominant diseases, if they pass on that dominant allele, they have the disease. Okay? Let me pull up, let's use a whiteboard right here because Punnett squares. Is it here? No. How do I get to whiteboard? Oh, can I just annotate and we do it that way? Oh no, it's gonna be, yeah. Okay. So Punnett squares are how you basically um, compare genetic disease, if you will, or odds of certain things being passed on. So heterozygous means you have one allele that is dominant and one allele that is recessive. So like they do a capital for dominant and then recessive for lowercase for recessive. So in this situation, if we're talking about a recessive disease, this person is a carrier. So we can say that's one parent. Let's say the other parent has the disease. They would have two recessive alleles. So in this situation, you put one parent here, one parent here. And what we would end up with, this one is a carrier because you're going to combine that dominant with the recessive. This one has disease. Man, this annotate's annoying because it wants to grab the line that I put. So annoying. This one is a carrier. This one has disease in this situation. So here it's 50% chance of having disease, 50% chance of carrier. For their next child, you would do the chart all over again. Okay, just because they had one kid, it doesn't mean it alters the ratios. No, it's a fresh situation. Now, if this is a dominant, things change. Okay, where is? Oh, you can't erase. Let's say it's a dominant disorder. Let's use the same people. Now this guy has the disease. This one does not. So any offspring that get that dominant allele will have the disease. So this is diseased, diseased, no disease, no disease. Okay? That's Punnett squares. So yeah, well, they have one there. That's fine. So autosomal recessive diseases you tend to see in childhood. So albinism, that's where they're lacking like melanin, PKU we talked about, cystic fibrosis we talked about, sickle cell. Typically you see them for very, very young age. Okay? Versus dominant disorders tend to kick in later in age. So remember I was saying like around 18, 19, that's where I started to see my hypercholesteremia. They start to ramp up. Same thing with type 2 diabetes is a dominant disorder. Okay. Huntington's disease. We talked about that. The dementia, neurofibromatosis. This is NF, and so they can have like hearing issues. They can have these growths in the brain. It can affect the brain. So it just depends on how severe it is. Marfan's, that's what it's referencing by the long fingers here. They're usually very tall. They develop what's called aortic dissection. It just randomly ruptures. Usually they have pectus excavatum. The sternum here points in instead of out. They actually suspect that Abraham Lincoln had Marfan's. And had he not been assassinated, if he had Marfan's, he probably would have died early on, like in his 40s or so, from aortic aneurysm or dissection, is what it's referred to. So, and they also have laxity in like joints, stuff like that. You can see in them. And then achondroplasia, which is dwarfism. So yeah, you'll see this early on, but they're still technically growing, and that's why it's a later life disease before it starts affecting. Okay. Sex-linked, so hemophilia, the bleeding, the muscular dystrophy, color blindness. Typically in males, there's a 50% increased risk of this because as a male, we only have one X. It's usually on the X chromosome. On the female, you have two X's, one turns off. So you have a chance of it turning off that bad one and you not expressing it as a female. So that's why we typically see higher numbers in males. Epigenetics. So this is talking about cancer, oncogenes. So we turn off on the CG, we methylate it on the DNA and we turn it off. If it unmethylates, the gene can be expressed. So when proto-oncogenes become oncogenes, so when you talk about like in HPV, that's what he does. He turns a proto-oncogene into an oncogene and now it becomes cancer. When it turns off tumor suppression genes, then these genes can be transcribed and translated cancer. Okay. So epigenetics, we methylate early on, that carries on cell to cell to cell down the road. And I think that's it. Oh, any questions?
No, I'm good.
Cool, cool, cool.
Yep, I'm good too.
So this is the plan. Every week I'll try and meet with you guys, lecture, we'll pick a day. I'd like to do it later in the week to give you guys time to get through the material.
Yeah.
But definitely stay on top of homeworks, do the homeworks, keep on track as if it's a regular class.
Okay.
And that way we don't fall behind.
Gotcha.
All right. Email me if you have
Cool.
questions. Enjoy your weekend.
Alrighty. Thank
Have
you
a good
so
weekend.
much.
See y'all.
All right. Bye.